I am Divya Shanmuvel and I am honored to present my research work which is conducted in Sinvesta titled as From Crabs to Crops and Electricity Nanochitosan as a biostimulant and bioelectric powerhouse for sustainable agriculture. So coming to the problem statement of my work is like modern agriculture faces two major challenges, declining soil fertility and increasing energy demands. Simultaneously, seafood industries generate tons of shell waste, especially from species like blue crab, which is Calinectus sapidus, which poses environmental disposal issues. Instead of treating this as waste, we saw it as an opportunity, an untapped resource that could be re-engineered into something valuable for both soil and energy system, which is called as nanochitosan. So the core of our research lies in the extraction and nanosizing of chitosan from blue crab shells. The shells undergo three steps of chemical process like demineralization, where the acid is used to remove the calcium carbonate, deproteinization with alkaline to eliminate the protein, and deacetylation with the concentrated sodium hydroxide to convert the chitin into chitosan by removing the acetyl group. So our nanochitosan had a particle size of approximately 33.37 nanometers with smooth spherical morphology, which is confirmed by some analysis, and it also showed a significantly high surface area, which is around 16.01 meter square per gram, and the pore volume, which making it for ideal for soil interactions. So we thoroughly characterized our chitosan using XRD, FTIR, DSC and BET analysis, and the thermal analysis showed strong thermal stability with sharp exothermic peak at around 335 degrees Celsius, which is indicating potentially durability in soil environment. The degree of deacetylation for nanochitosan has reached up to 91.46%, which is significantly higher than the commercial chitosan, which is used here for the sigma aldrich. And this directly translates to better solubility and bioactivity in agricultural settings. When applied to soil, this nanochitosan, which is extracted, has showed powerful biostimulant effects like moisture content, which is increased up to 22% in control, but the treated soil has 39%. Coming to water holding capacity, which is improved to 84%, and the total nitrogen and carbon level has increased by 2.4 and 1.2.4 to 1.4 times higher comparing with the control one. Coming to the plant growth trials, here the plant which I chosen is Solanum lycopersicum, which is a tomato plant, has showed a remarkable result when it is grown in the soil treated with chitosan. So the germination percentage is also increased. Like in control, it's only 22 percentage, but in nanochitosan treated soil, the germination percentage was around 87%. The plant height is around 30.6 centimeter, and the dry biomass is nearly highly increased comparing with the control one, and there was also a notable increase in flowering and stem diameter comparing with the control and the treated. So based on these results, it is clearly shows and demonstrated that the nanochitosan not only improves the physical soil properties but also enhance the nutrient availability and the plant development. So next, coming to the phytohormonal and phytochemical metabolomic responses, we used LCMS analysis. Based on this, we identified several bioactive components which is present in the tomato plant treated with nanochitosan has shown a tremendous improved quality, which is including like phytohormones such as hasmonic acid, abscisic acid, and indole 3 acrylic acid, phytochemicals like chlorogenic acid, ferulic acid, and trans-lycopene, and the steroid alkaloids like tomatidine. All these are shown in improved peak comparing with the controlled one. And these compounds play a vital role in stress resistance, root growth development, and overall plant growth development. Notably, you can see the Brix percentage, which is shows the levels of sugar content which is present in the fruit has also increased in the tomato which is grown in the plant treated with chitosan. So this indicates that enhanced sugar content and the fruit quality is also improved when the plant is grown with the soil which is treated with nanochitosan. Then coming to bioelectricity generation, here comes the exciting part, the energy generation. by fixing an electrode in towards the rhizobium zone of the plant where the nanochitosan treated plants used. So we observed a bioelectric activity where the maximum voltage reached up to 1.503 voltage and the power density peaked up to 0.12 millivolt per centimeter cube. So this opens the door for integrating plant microbial fuel cells into farming system, allowing for micro energy harvesting in greenhouses, smart sensors or autonomous irrigation units. And not only the electricity generation or plant growth development, there also notably noticed that soil microbial community. The microbial analysis revealed that nanochitosan enriched soil biodiversity, and also we identified some bacterial strains like Glutamicibacter, Nocardioides, Bacillus asahi, and Pedobacter. Presence of these bacterial strains comparing with the control one, these bacterial strains have shown enormous colonies in the treated soil, especially Glutamicibacter is only present in the treated soil treated with chitosan. It was absent in the control. So these bacteria are linked to nitrogen fixation, phosphate solubilization and natural disease resistance. And the heat map which is provided here, the data showed significantly enhanced microbial activity which is providing a nanochitosan role as a sustainable soil amendment. So based on these work, I like to conclude that my research demonstrated that the nanochitosan derived from crab shell is a multifunctional agent. It enriches the soil, promotes healthier plant and also a productive plant which supports the beneficial microbial life in the soil, and also it even generate electricity. So in one work, you can do a three possible outcome from a project, and this innovation will give a bridges marine waste recycling, agricultural sustainability and renewable bioenergy in one comprehensive solution. And I believe that this work could influence the green farming practices, especially in resource limited and climate sensitive regions. And finally, I would like to acknowledge my thesis directors, Dr. Omar Solorsa and Dr. Satish Kumar Kamaraj. And also, I would like to thank CONACYT for giving me a doctoral fellowship scholarship for my project, and also Sinvesta for the doctoral support. So I would like to thank all my companions and friends and colleagues, those who help in this work, and a special thanks to National Health Laboratory, Unam Istakala, for their support in doing the soil microbe analysis. So thank you so much for your time, and I welcome if you have any questions or feedback, you can give in a comment section. Thank you so much.